Chapter 11, Part 10. So we're going to move on to the synapse. So the synapse is the junction between two cells. It can be between a neuron and a neuron, or a neuron and a muscle cell or gland. So this is the site where action potentials in one cell cause an action potential in another cell. There are the types of cells in a synapse are basically the presynaptic cell and the postsynaptic cell. So the presynaptic cell is the cell that transmits the signal towards the synapse. It's where it's coming from and going to, basically. And then the postsynaptic cell is the target cell receiving the signal. So there's two types of synapses, electrical and chemical. So a chemical synapse, the components are the presynaptic terminal, the synaptic cleft, and the postsynaptic membrane. So neurotransmitters are released by an action potential in the presynaptic terminal. So meaning, and I want to clarify that, the neurotransmitter is released because of an action potential that happens in the presynaptic terminal, and those neurotransmitters are released into the synaptic cleft. Okay, so remember that we have vesicles in the end of a neuron, into the terminal end. They're called synaptic vesicles, and those synaptic vesicles they're like little storage containers for neurotransmitters. And the one that we will talk about mostly in this chapter is the acetylcholine, which you should be familiar with from the muscle system. So the synaptic vesicles, the action potential causes potassium to enter the cell, and that will force the synaptic vesicles to release the neurotransmitter into the synaptic cleft. The neurotransmitter will then diffuse across that synapse, and the postsynaptic membrane will have receptors waiting for that particular neurotransmitter. And when the neurotransmitter binds to those receptors, there are ligand or chemical gated ion channels that will open, and that sometimes it's going to cause sodium to be released or enter or whatever, depends on where we're at. But we're going to talk about that a little bit more coming up. So this is a chemical synapse. This is a review for muscles, like specifically when we talked about the events of the neuromuscular junction, it's just like that. So you can see this looks familiar. You have your terminal end, or the presynaptic terminal. It's basically the end of the neuron, okay? The axon would be coming from here. Here comes the action potential. That action potential changes the membrane potential enough to cause these voltage-gated calcium channels to open, because a voltage-gated channel is triggered by a change in voltage, which would be an action potential. And when those calcium channels open, then calcium is going to kind of rush in, and that entrance of calcium into that end bulb, into this end of the neuron, will force these vesicles to release their neurotransmitter into the cleft, which we're talking about here is the acetylcholine. And then that acetylcholine will bind to receptors in the postsynaptic membranes. This is supposed to be a membrane of the next cell, right? This whole thing is the synapse, and here is the synaptic cleft, the gap in between. So once those vesicles empty their contents into that synaptic cleft, they will, which are the neurotransmitters, those neurotransmitters will attach to these sodium channels, causing them to open, allowing for sodium to either leave or enter, depending on which way. And this looks like it's entering, because this is kind of an enlargement. This is an enlargement of this section right here. And it's showing that sodium is entering into the cell based on those neurotransmitters opening the gates. So there must be more sodium out here than there is inside. So sodium is going to flow down its concentration gradient into the cell. So that, again, is kind of a review from the events at the neuromuscular junction from the muscles. So anyway, when that happens, once those gates open and allows for the sodium to enter, that basically the neurotransmitter causes a change in the membrane potential, allowing for the signal to travel along the cell membrane, and then to the next, either to the next cell or causing something to happen, whether it be the muscles contracting or the gland to secrete something. So anyway, again, this is basically something you've kind of heard already based on what we talked about in muscles. So one other topic that comes up in this chapter that I don't jump up and down about, but you do have to know how the neurotransmitter is removed from the synapse, so or from the cleft. So acetylcholine is removed by acetylcholine esterase. Acetylcholine is removed from the cleft by acetylcholine esterase. Actually, there's a couple of different ways that acetylcholine is removed. Acetylcholine can diffuse away, and then some of the acetylcholine will be broken down by acetylcholine esterase, which basically, I don't really care what acetylcholine is broken down into, but the parts are then recycled back into the presynaptic neuron. So basically, once the neurotransmitter acetylcholine in this cleft is done doing its job, it will, number one, sometimes diffuse away, and other times it will be broken down by acetylcholine esterase, and then there are special pumps in the ends of the neurons that are not shown here that will basically uptake those parts of the acetylcholine the acetylcholine's broken down into smaller parts, and then those parts are taken up back into the synaptic end bulb, and then the parts are then remanufactured into acetylcholine, repackaged into vesicles, and ready to go again. So if we're talking about norepinephrine, that is recycled within the presynaptic neuron or diffuses away from the synapse. Monoamine oxidase is the enzyme that will help to break it down. So I don't really focus so much on the norepinephrine. You're going to hear me mostly talk about acetylcholine. So removal of the neurotransmitter from the synaptic cleft. So basically, I already kind of went over this, and you can read through this. Again, there's a couple different ways it happens, but this book is pretty concise on what it's telling you here. And then here is the removal of norepinephrine. Again, I'm not going to make a big distinction between acetylcholine removal and norepinephrine removal. If you know that, generally speaking, if it's broken down, those parts are recycled, or the entire neurotransmitter is recycled. All right, so receptor molecules in the synapses. So specificity is referring to the neurotransmitter only fits into one receptor. Okay, those receptors are very picky. The neurotransmitter is only going to link up to the receptor that matches it perfectly. And this causes the neurotransmitter to affect only the cells that have those receptors for that particular neurotransmitter. So a neurotransmitter, say norepinephrine, isn't going to work on a receptor that only works for acetylcholine. So neurotransmitters can be excitable in some cells and inhibitory in other cells. So that means neurotransmitters can either excite or inhibit. They can either cause something to happen or inhibit something from happening. So some neurotransmitters, for example, norepinephrine, can also attach to this presynaptic terminal and modulate its own release. So it's basically kind of like a built-in negative feedback system. So neurotransmitters and neuromodulators. So chemical messengers secreted by neurons are basically neurotransmitters, and neurons can secrete more than one type, and there's about 100 different types at least. So there are major chemical classes of neurotransmitters. The most understood and most studied neurotransmitter is acetylcholine. And then you can see here, there's a bunch of different other ones here. So be familiar with these, that these are the major chemical classes of neurotransmitters. And I know that there's a table in your book that outlines these as well. So responses at the postsynaptic cells can be excitatory and inhibitory. So an excitatory postsynaptic potential, EPSP, basically depolarization occurs and the response is stimulatory. And then let me move to the next one. The inhibitory postsynaptic potential, IPSP, is basically hyperpolarization and the response is inhibitory. So it decreases the likelihood of an action potential by moving the membrane potential farther from the threshold. So when we want something to stop or not happen, this is what will happen. When the body doesn't want something to happen, it's an IPSP. So know the difference between an EPSP and an IPSP. So spatial and temporal summation. So a single postsynaptic potential is not sufficient to reach threshold. Many postsynaptic potentials combine in what is referred to as summation at the trigger zone. And if threshold is reached, an action potential is triggered. There are two types of summation, spatial and temporal. So spatial is basically action potentials 1 and 2 cause the production of a graded potential at two different dendrites. So an action potential will come in here, an action potential will come in here, and eventually, if enough come in, then that's summation from in regard to spatial summation. So action potentials are coming in from different dendrites. In temporal summation, it's an action potential coming in repeatedly from the same dendrite. So two action potentials arrive in close succession at the presynaptic membrane, and it's actually two or more action potentials. So combined spatial and temporal summation, an action potential is produced at the trigger zone when the graded potential is produced as a result of EPSPs and IPSPs summate to reach threshold. So basically, if enough excitatory come in to outweigh the inhibitory, then an action potential will actually happen. So I'm going to stop here and pick up on these last two slides. Actually, no, I have time to finish this. So there are different pathways, neuronal pathways and circuits. So the organization of neurons in the CNS varies in complexity. So there are convergent pathways, divergent pathways, reverberating circuit, and parallel after discharge circuit. And so there's some descriptions here, but I like to go by the pictures for this. This to me makes more sense. So when we're talking about a convergent pathway, it's basically we have a whole bunch of inputs converging into a single output. Okay, so we have all these inputs coming to one neuron. A divergent pathway is the opposite. We have one input that goes to 2, that goes to 4. So it diverges out. This converges into 1, this diverges out into multiple. Reverberating is basically like it keeps stimulating the original few neurons. So it kind of goes in this circular pattern to keep stimulating over and over again. And then the parallel after discharge is it starts in one, goes out into multiple sections, and then finds its way back into a single neuron. So that is it for chapter 11, finally. I look forward to you hearing all about the next chapter.